Metabolism in insects. Substances absorbed through the gut wall seldom remain unchanged in the hemolymph for any length of time, but are quickly converted into other compounds. Metabolism comprises all of the chemical reactions that occur in living organism. It includes anabolism and catabolism. Anabolic reactions include, for example, the formation of structural proteins or enzymes from amino acids and the formation of simple sugars of polysaccharides that serve as an energy store. Many catabolic reactions have evolved for the specific purpose of producing the large quantities of energy required by the organism for performance of work. The metabolism of insects generally resembles that of mammals, details of which can be found in standard biological texts. The present account, therefore, will be largely comparative in nature. Sites of metabolism. Chemical reactions are carried out by all living cells, though they are usually limited in number and of course are related to the specific function of the cell in which they occur. For example, in midgut epithelial cells, metabolism is directed largely towards synthesis of specific proteins, the enzymes used in digestion. Metabolism in muscle cells is specifically concerned with production of large amounts of energy in the form of ATP for the contraction process. In epidermal cells, reactions leading to the production of chitin and certain proteins, the components of cuticle, are predominant. Certain tissues, however, are not so specialized, and in them a multitude of biochemical reactions involving the three major raw materials are carried out. In vertebrates, the liver performs these multiple functions. The analogous tissue in insects is the fat body. Fat body. The fat body is derived during embryogenesis from the mesodermal wall of the ophthalmic cavities. In other words, it is initially a segmentally arranged tissue. Through this becomes obscured as the hemocoel develops. Nevertheless, the contrary to what a casual examination may suggest, the fat body does have a definite arrangement in the hemocoel characteristic of the species. Typically, there are subepidermal and previsceral layers of fat body, plus sheets or cords of cells occur in other specific locations. Thus, the fat body presents a large surface area to the hemolymph, allowing the rapid exchange of metabolites. Contrary to what was thought originally, the fat body is not a single uniform tissue. Not only are there regional and species specific differences, but also differences between larval and adult fat body as well as in fat body cell types have been reported. Groups of cells of each type are located in specific positions throughout the body. It should be stressed, however, there is evidence for the functions of these many histotypes. The fat body is composed mainly of cells called trophocytes, though in some species urate cells and rhomocytocytes also can be seen scattered throughout the tissue. In embryos, early post-embryonic stages and starved insects, the individual trophocytes are easily distinguishable. Their nucleus is rounded and their cytoplasm contains few inclusions. As such, they closely resemble hemocytes, with which they probably have a close phylogenetic relationship. In later larval stages and adults, the trophocytes enlarge and become vacuolated. The vacuoles contain reservoirs of fat, protein, and glycogen. The trophocyte nuclei are proportionately large and frequently become elongate and much branched. During metamorphosis in endopterygotes, the reserve are liberated into the hemolymph. In some Diptera and Hemiptera, the majority of trophocytes also disintegrate at this time, and the fat body appears to be completely reformed in the adult form, the few cells that remain. In a number of species, uric acid accumulates in large quantities in specific cells, the urocytes, within the fat body. In cockroaches, the urocytes surrounded Mesito Sites, and there is circumstantial evidence to suggest that the bacteria are intimately involved in the synthesis and utilization of the uric acid. Mesito Sites. Mesito Sites or bacteriocytes are found in widely different groups of insects. Though in common, these have nutritionally poor or unbalanced diets such as wood ants, circumambitids, and anobidid beetle larvae, phloem sap feeders like hepids, plant hoppers, mealybugs, and blood feeders, bedbugs, tsetse flies, sucking lice. Mesitocytes are generally stated to be specialized fat body cells. However, in the pea aphid Acrithosepium pisum, some mesitocytes originate from nucleic near the posterior end of the embryo, while others may have their own origin as nuclei in the middle of the embryonic central syncytium. Usually, the mesitocytes from specific structures known as mesitomes or bacteriomes, distinct from the fat body. The mesitocytes contain symbiotic bacteria for which various roles have been proposed. The mesitocyte system of aphids is practically well studied. The primary bacterial symbiont is Buchnera aphidicola, which is inherited transferrially from mother to daughters via the ovary. The number of mesitocytes in the host is fixed at birth, though the cells grow to about 4 times their original size during larval development as the bacteria multiply. For aphids, there is a strong evidence that the bacteria supply the host with essential amino acids. The bacteria may also participate in nitrogen recycling and upgrading as suggested above for cockroaches. In aphids, the evidence is against a role for the bacteria in the synthesis of vitamins and lipids. However, this possibility cannot be ruled out for other insect groups. Curiously, and contrary to what is generally assumed, there is no known benefit to the bacteria from this association. Carbohydrate metabolism. As in other animals, simple sugars provide a readily available substrate that can be oxidized for production of energy. However, in contrast to vertebrates, where glucose in the blood is the sugar of importance as an energy source, in insects, hemolymph glucose and other monosaccharides usually are present only in minimal amounts. An exception to this statement is the worker honey bee, whose hemolymph glucose may reach a concentration of almost 3 grams per 100 ml and is used as the energy source during flight. In most insects, a disaccharide trihalase is the immediate energy source. Trihalase consists of two glucose molecules joined through an alpha 1 linkage. Its level in the hemolymph is constant and in a state of dynamic equilibrium with glycogen stored in the fat body. In this respect, therefore, the situation is comparable to that in vertebrates whose blood glucose level is in equilibrium with liver glycogen. The similarity goes further. Just as the conversion of liver glycogen to blood glucose is promoted by the hormone glycogen, which stimulates glycogen phosphorylase activity in insect fat body, the formation of trihalase from glycogen is prompted by a hyperglycemic hormone released from the corpora cardiaca. This hormone activates the phosphorylase, which removes a glucose unit from the glycogen. Because of its high polar polyhydroxyl nature, trihalase does not easily penetrate the muscle cell membrane. Therefore, before it can be oxidized by muscle, it must first be converted into glucose by a hydrolyzing enzyme trihalase present in the muscle cell membrane. Hemolymph trihalase is also the source of the glucose that is converted by epidermal cells into acetyl glucose amine during production of the nitrogenous polysaccharide chitin. Glycogen is an important reserve substance in almost all insects and is found in high concentration in fat body, with similar amounts in muscle, especially flight muscle, and sometimes the midgut epithelium. In the mature bee larva, for example, glycogen makes up about 1/3 of the dry weight, which is produced principally from glucose and other monosaccharides absorbed from the gut following digestion. In some insects, glycogen may also be synthesized from amino acids. As noted above, fat body glycogen is used to maintain a constant level of trihalose in the hemolymph. The glycerol produced as an antifreeze in the hemolymph of some insects that must withstand extremely low winter temperatures is also derived from fat body glycogen. Glycogen in muscle is used directly as an energy source, being degraded as in mammalian tissue via the glycolytic pathway, Krebs cycle and respiratory chain with resultant production of ATP. Glycogen also is a significant component of the yolk in the eggs of some insects. Its use as an energy source in this situation is, however, secondary to its importance as a provider of glucose units for chitin synthesis in the developing embryo. As in vertebrates, the major functions of the pentose cycle in insects are production of reducing equivalents as NADP that are used, for example, in lipid synthesis, the production of 5 carbon sugars for nucleic acid synthesis. In addition, though its ability to interconvert sugars containing from 3 to 7 carbon atoms, the pentose cycle can change unusual sugars produced during digestion into 6 carbon derivatives and hence into glycogen. Lipid metabolism. For most insects, fat stored in the fat body are the primary energy reserve, and like those of other animals are mostly triglycerides. They may be formed directly by combination of the fatty acids and glycerol produced during digestion or from amino acids and simple sugars. Typically, fat is stored throughout the juvenile period, especially in endopterygotes, where at metamorphosis it may make up between one third and one half of the dry weight of an insect. Large amounts of fat also accumulate in the egg during vitellogenesis. Fats are used as energy source during long-term energy requiring events. For example, embryogenesis, metamorphosis, starvation and sustained flight. On a weight for weight basis, fats contain twice as much energy as carbohydrates. They are therefore more economical to store. In addition to the fats just described, which serve slowly as energy reserves or source of carbon, many other lipids having structural or metabolic functions occur in insects. Waxes are mixtures of long chain alcohols or acids, their esters and paraffins. The number of carbon atoms that form the chain ranges between 12 and 36, and both unsaturated and saturated compounds have been identified. It seems that the various components of wax are synthesized from fatty acid precursors. The paraffins and possibly some acids are produced by Amino sites, whereas alcohol and ester synthesis occur in the fat body. Compound lipids are fatty acids combined with a variety of organic or inorganic residues, for example, carbohydrates, nitrogenous bases, amino acids, phosphate and sulfate. The metabolism of these lipids in insects is for the most part poorly known. Certain of them, for example, choline, a phospholipid, cannot be synthesized by insects and must be included in the diet. Likewise, sterols are essential components of the diet in almost all insects. Amino acid metabolism. In growing insects, a large proportion of the amino acids that result from digestion is used directly in the formation of new tissue proteins, both structural and metabolic. Within the fat body especially, but also in other tissues, a variety of transminations also occur. That is, the amino group from an amino acid can be transformed to a keto acid to form a new amino acid. Such transminations are especially important when an insect's diet contains insufficient amounts of particular amino acid. As in vertebrates, not all amino acids can be synthesized in insect tissue. Those that cannot must be included in the diet are provided by symbiotic microorganisms. During starvation or when present in excess, amino acids may undergo oxidative deamination within the fat body, resulting in the formation of the corresponding keto acids. The latter can then be further oxidized via the Krebs cycle and respiratory chain to provide energy or may be converted into carbohydrate or fat reserves. The ammonia produced during deamination is normally converted into uric acid. The fat body is an important site of hemolymph protein synthesis, especially in the late juvenile stages of endopterygotes and in adult female insects. Storage hexamers have been characterized from more than 20 species in 6 orders, mainly Diptera and Lepidoptera. They reach very high concentrations in the hemolymph just before metamorphosis and are effectively serving as a store of amino acids for use in adult tissue and protein formation. If stored individually, the amino acids would create a severe osmotic problem for the insect. In silk moth, bombyx, caterpillars, for example, the hemolymph protein concentration increases 6-fold from the 4th to 5th instar. In preparation for spinning the cocoon, metamorphosis and egg production. When the adult emerges, the concentration of fallen to about 1/3 the value at pupation. During sexual maturation in the females of many species, the fat body produces vitellogenesis, female specific proteins. These proteins, whose synthesis is regulated by juvenile hormone or ecdysone, are accumulated in large amounts by the developing oocytes. In female insects that do not feed as adults, example bombyx, the storage hexamers are the source of the amino acids for vitellogenin production, which occurs during the ferret adult stage. In the males of some species, the fat body produces proteins that are accumulated by the accessory reproductive gland, probably for use in spermatophore production. Lipoproteins are another important group of hemolymph proteins synthesized in the fat body. These often very large molecules are important in the absorption and transport of lipid and additionally serves as coagulants in hemolymph clotting. Coming to the summary, typically saliva lubricates and initiates digestion of the food. However, it may include compounds that act indirectly to facilitate food uptake and digestion, or that have functions unrelated to feeding. The gut includes 3 primary subdivisions: foregut, midgut, and hindgut, and these are typically differentiated into regions of differing functions. The foregut is concerned with storage and trituration of food, the midgut with digestion and absorption of small organic molecules, and the hindgut with absorption of water and ions. Though some absorption of small organic molecules may occur across the hindgut wall, especially in insects with symbiotic microorganisms in their hindgut. The digestive enzymes produced match qualitatively and quantitatively the normal composition of the diet. The enzymes have low specificity, enabling an insect to digest a variety of molecules of a given type, or may be highly specific. For example, when a species feeds slowly on a particular food, gut fluid is buffered within a narrow pH range to facilitate digestion and absorption. Enzymes are released as soon as they are synthesized. Synthesis is regulated so that an appropriate amount of enzyme is produced for the food consumed. Microorganisms in the gut may be important in digestion, especially in wood-eating species, where they degraded cellulose. Absorption of digestion products occurs mostly in the anterior midgut. It is generally a passive process, though carrier molecules may be used to facilitate the process. the rate at which sugars are absorbed is linked to the rate at which they are converted to trihalose and hence glycogen. Lipid absorption is generally slow, with the lipids being converted into di and triglycerides as they move through the midgut epithelium. Amino acid absorption may be preceded by absorption of water across the midgut wall to produce a favorable gradient for diffusion. However, facilitated diffusion and active transport systems are used for some amino acids in some species. Thank you.